Yes, good afternoon. Kindly be seated. Let us start the remaining presentation. It's on for John. Okay. Yes, I think Thirumalai will start the presentation. இல்ல உங்களுக்கு இல்ல. We'll start because you are discussing now. That's why. Dr. Priyank will start the presentation. Next speaker. Dr. Jean, I think the technical problem is resolved now.
I hope so.
Yeah.
We will see. The
Okay.
solution was to switch to an Indian computer who seems to be
Okay.
more compatible with the Indian video projector.
Okay.
So,
But still, your time is 20 minutes. I think you kindly bear with us because we have to accommodate the speakers as well. Since you are linked with, I think, some online presentation, we can give 5 minutes more extra. Kindly proceed.
okay. Thank you very much, Mr. Chairman, and thank you for giving me the opportunity to give this presentation. I hope you had a nice lunch break. So the talk that I will share with you will deal with first modeling and then agroecological protection. So we saw already in the morning 2 presentations about modeling. Yeah, thank you. And so I think it will resume. And the other objective is to present 2 innovative tools that allows for non-specialists of modeling to be able to design their own model, to share them and to use them. And so I hope it will be a possibility to generate collaboration. And maybe for the overall audience, I think it's good if everybody cuts the telephone, because I noticed this morning there were many phone calls, so it's good if everybody put on the mute, please. Okay, so let's start.
Kindly be attentive because with a lot of difficulties we got the good speakers. Kindly put off your mobile phone. If it is urgent, you can go out to interact. Yes, please.
Okay. So first of all, why modeling? So I think we already had some explanation by Frédéric, but I try to sum up some idea because the question is general. It's not specific to plant protection or to agronomy even. It's used in all type of activities for human beings. So one important goal of modeling is to better understand the behavior of a given system. Okay, very simple. Another goal of modeling is to gather existing knowledge and identify knowledge gap. So for research, it's very important because once you have expert knowledge or knowledge from the literature, it's a way to gather in one place the information, like what you saw either for potato forecast or also for cyst nematode in the last presentation. Then we have another important use of model is to support decision making, to predict the behavior of a system under a given management. So for instance, if you want to know when to apply a fungicide, it's important to have a model to know, okay, it's now that the disease is really increasing very fast and you should apply your fungicide. Then another objective of developing model is to teach and to train. It's very easy when you have students to explain a system and to make them use a model or to present the model, and it's a very efficient way for teaching. And at last, it's also a very efficient tool to communicate. In the past, we had designed some models with video outputs, and some extensionists use them in order to go to farmer place and explain some consequences of their cropping practices. So to me, it's the 5 main reasons of why we are using model. So just briefly, what are the key stages of a modeling project? The first stage is very important, is called the problem shaping. What is the question? It seems to be obvious, but it's not always the case, so we have to really think deeply about that. Who are the final end users? And if at all possible, it's very nice if you can include the end users in the process of model development. And also you have to think about what are the available sources of knowledge. Then come the phase of conceptual modeling. I don't know if you understand what I mean by conceptual. You just, when you go on a white paper and you draw arrows and blocks and you say, okay, this variable should play a role in this system, and it's a very important phase of a modeling project. Then once you design a conceptual model, you have to translate this conceptual model into a simulation model. So usually it's done with mathematics, but you can do many types of options. You can do partial equations, you can do mechanistic equations with derivatives and so on. So it's an affinity. You can use statistical approaches. It's a whole world. Then before providing advices to farmers, it's usually a good idea to have some estimate of the quality of prediction of the model that you're using. Because if your model does not predict correctly the reality, you might provide bad advices to farmers. So it's an important phase. Then it's a little bit technical. I don't know if it rings a bell to you. I'm sure for Frédéric it does. So it's to perform sensitivity and uncertainty analysis in order to understand what is the impact of the uncertainty of your input variables and what are the most important parameters in your model. Then you can re-estimate or estimate the parameters of the model. Then you can launch and run simulations. And then once you're happy with your model, you can disseminate, share it. So it's the main phase of a modeling project. And of course, it's not linear. You have a loop, because all these stages are in interactions, and you always go back to your model. And it's also a nice way to interact between data sets provided by experiments or observations and the model being developed. So I talk about modeling, but I did not explain what is a model. So there is an agreed general definition. It's very simple. A model is a simplified representation of reality. So you see, with that definition, okay, it can include mathematical models, but also if I don't know, I take some painting and I try to paint the audience now, I will try to represent in a simplified way the reality of the room. So it's also a model, okay? So using that, I will try to show you some ideas about modeling using a famous painting from a French painter of the 19th century. So I don't know if you have ever seen this painting. It's called The Gleaners by Millet. Do you know what are the gleaners? You know, it's people who go on the field and to pick up the grains that were left after harvest. So here you have this painting, and you have three ladies who pick up grains after harvest. So this is one vision of agriculture. The idea of the painter was to show how hard was the life in the countryside in the 19th century. So you see the bodies, they are in a painful position, and you see the colors are darker in the front, and in the back you can see big harvest. And so it's a way to show the differences between the rich farmers who own their farm and workers who are not rich and who has to take the food left on the soil in order to eat. So what I want to stress is that, so okay, if we agree with the definition, this is a model of reality. But the painter used a technique here using painting, But some years before, he used another technique. He used some acid on copper, and he made the same idea. So he made another model using another technique. So it's just to show you that models can be designed using different techniques. So it's one first idea. And there is another idea I would like to express, is that this is another painting from another painter. Okay, the colors are not so great on the screen, but usually the colors are brighter, and you can see it's another vision of agriculture, because here, you know, the ladies, they come back home with a lot of grains left at soil surface, and the bodies are straight, and it's just to show you that modeling is also a point of view. You can see that it's the same scene, gleaners, and here it's a point of view, and here it's another point of view. So it means that if you are interested to model a system, you will have many ways of modeling it, either by having different point of view, and this is very important when you consider conceptual modeling, or using different techniques. Okay, so it's two things. So imagine that Millet, if he was not a painter, and actually if he had been a modeler, instead of painting, maybe he would have said, okay, I will think about making a conceptual model. So I just, to exemplify the idea, I designed for him a conceptual model of gleaners. So I say, okay, what I want to predict is the total biomass collected on the field by my 3 gleaners. And then I stop and I think what will be important to predict that. Well, it will depend, of course, of the number of gleaners in the field. It will also depend on the field area. What will be the working time of the gleaners? Also, what are the number of grains left at soil surface after harvest? And this will define biomass collected by gleaner per unit time and per unit area. And then it depends also on the physical thickness of a gleaner. So if you are not familiar with that, here is maybe, if it's the first time you see a conceptual model. So what I want to show you now is how to translate a conceptual model into a simulation model. So here is an example. I can simply state an equation. I will say that the biomass that is collected in the field will depend on the number of the gleaners multiplied by a function that will describe the quantity of grains collected by the gleaners. And it will depend on the physical fitness of the gleaners and the density of grains. And in my equation, I say the bigger the field, the bigger will be the grain collected. So I multiply by S. And also, this will be the working time, delta T. So it's very simple. I had my conceptual model, and now I made. So this is called a static model, because you don't have the impact of the time. So this is the name of the second painter, Breton. Okay, but like I said, here you remember, I can use different techniques. So I say, okay, this model is not bad, but I would like to know at each hour of the day what is the biomass collected. So then I will introduce a dynamic model. So it's easy, I just integrate as a function of time the same equation, because the number of gleaners and the field surface does not change over time. I can move it from the integration. So you see, one conceptual model, I already made 2 different models, one static, one dynamic. And why not? We can have fun. A third example, I can decide to design a spatially explicit representation of the grain distribution. So it's the blue dots in the field here. And so if there is no spatial distribution, I can call that a 2-dimensional isotropic model. And I can say that, okay, the gleaners are represented by individual based models who are not coordinated and who follow a Brownian trajectory. So it's some assumption that I'm making. And then I can make some stochastic simulation using some program. So just, okay. One conceptual model, 3 different models. Okay, so now if we go back to agronomy, I try to stress the complexity of our system. Here, in this purple rectangle, you have a representation of a field. So in a field, you will find soil with physical and chemical properties, and crop. It can be one monocrop, or it can be intercrops, or it can be crop with non-harvested plants also. And this is under the influence of all the biological components. So you will have pests. And in my word, when I say pests, it's not only animal pests, but also plant pathogens and weeds, okay, and beneficials or other living organisms. And then you have microclimate, both for the soil and the crop. But this system, you know, it's already quite complex. In addition, it's open, because it has some influences from the landscape. Some pests can come from the field of your neighbor, if you're a farmer, and it's also under the influence of weather, okay? And what is very important is the cropping practices applied to the field that will have impact on all these interactions. So according to a set of social, economic, and environmental drivers, the farmers will have some perception and cognition that will lead to cropping practices, and then it will end up to have a vector of social, environmental, and economic performances. So it's a very simplified representation of what's going on in an agroecosystem, but you can see that there are many arrows, many boxes, and so on. And models can help handle high level of complexity. So if we go back to crop protection, there is an important concept called agroecological crop protection. So we wrote 2 books about it, one in English, which is presented here, and we provided a definition. If you are interested by words in the field of agroecology, in my lab we developed this dictionary, and there is a 1-page definition of ACP. And the basic message is that ACP, agroecological crop protection, is the declination of agroecology to crop protection. And we also wrote a paper. You see there are many authors in order to propose the basic principle of ACP. And there is also an online training tool in order to help students to understand what is agroecological crop protection. So it takes about 8 hours to perform the training session. I have to introduce another concept. I don't know if you're familiar with that in India, is the concept of ESR. So efficiency, substitution, redesign. Efficiency, it's when you optimize pesticide use. So it can be just modifying the nozzle that you're using for spraying, or it can be using decision support system to decide when and which pesticide use. Substitution is when you replace a pesticide use by, for instance, a biocontrol application, or if you have some physical protection, it will be substitution. And redesign is when you change the cropping system. So you change the rotation, or you introduce new crops. So it's deeper changes in your system. So I need that because I used it in order to make a typology of the different objectives that we can have for modeling in crop protection. We can design farming warning systems, like we saw, so it deals with the efficiency or the substitution. We can design decision support system for chemicals or biological control. We can use that to design agroecosystem less susceptible to pests. Design strategy to preserve cultivar resistances, so you go for an understanding of the genetic structure of the pest population. You can also think about designing landscape management strategy to limit or to control the pest population. You can also use that if you have an architectural model to change the control strategy through changing the microclimate through the architecture management. You can use models to design ideotypes. You can perform yield losses analysis. You can perform analysis to analyze invasive species. You can work on analyzing the effect of climate change. You can assess multi-performances of ACP strategies in a multi-dimensional way. You can use modeling to design sampling strategies for monitoring, optimizing the resources you have to do that. And you know, it's just examples, but there are many things that can be done with models. Okay. Now let's think about where we have knowledge to produce some advices or information for farmers. The first source of knowledge are experiments. Either it can be in the field or in the greenhouse or in the lab, but you set up a protocol, you set up an experimental plan, and you have data set. So it's a source, and it's a primary source of information. Another important way of producing knowledge in science is to observe. In some scientific fields, you cannot perform experiments. If you are studying planets, you cannot play with that, and you cannot perform experiments. So you can just observe. And in the field of crop protection or agronomy, it's the same. You can go in commercial fields and ask the farmer what was his practices and just observe what's going on. And then you have what I call secondary sources of knowledge. You can read the scientific and technical literature, usually based on experiments and or observations. You can use existing models that also are a source of information. And also you have something that each brain of the room has, it's expert knowledge, because you went to the field, you also read many things, and each actor has some knowledge. So a farmer has its own expert knowledge. An advisor has also his expert knowledge, and such as also researchers. So now how to integrate all this knowledge. So among the audience, who is found who likes to do mathematics? Thank you, Frédéric. So everybody can leave. No, it's the opposite. Because usually when working in the field of agronomy or biology, people are not really familiar with equations, mathematics. And about 10 or 15 years ago, I had a colleague who was actually a teacher, a professor in crop protection for 20 years. And she asked me if I could be her supervisor, which I was very happy about. And so we started to work on a PhD thesis. But I realized that first, she was not at all interested by mathematics. And second, it was not her thing at all. So it's how we come up with the idea of designing models without using any equation. And it was very successful because many teams were able to use all that type of knowledge already existing, including expert knowledge, in order to design. So it's a very easy way to design models. So it's what we are doing is called injury profile simulator, because we wanted not only to address to one pest, but to be able to model all type of diseases, animal pests and weeds. So the basic idea is that the vector of injury profiles, injury severities, only depends on three components: soil and weather characteristic, the cropping practices, and the landscape. So we apply that on wheat production. You can see here 13 pictures, which represent 13 pests on wheat. But after we did another PhD on sunflower, we progressed more on the modeling methodology. It was used in 4 European projects, which are listed there, and you know we applied it to wheat or to rust on wheat and even to intercropping. And several teams, mostly French teams, use it for other crops, such as apple tree production, also chariots, pest on mango trees, and also we helped with colleagues from CIRAD in Costa Rica to design a coffee rust decision support system. And also we use this modeling platform for various trainings. So this one, a summer school in Italy that we adapted to Southeast Asia. And so these are some pictures. So I will just explain one presentation.
Yes, please.
So this is a chow chow, and it's a first collaboration with an Indian colleague. So thank you, Kavita. She took this picture in Uti yesterday. And I thought about this picture because we designed a model on Chayot. So we designed it in Réunion Island. So it's a little bit closer than mainland France. It's about 5,000 kilometers from France, whereas from Paris to New Delhi, it's 7,000 kilometers. So you have some pictures of Réunion Island. This is the Chayot there. And in this island, there were a big problem with fruit flies. And this is why we came up with the idea of designing the model Ipsim Chayot. I will not go too much into detail, but it's a simple decision tree that you perform using expert knowledge because there were not so many articles available. And this is what we call a confusion matrix, which represents the quality of prediction of the model. So if you're interested, there is a paper, and now I will try to switch to an online platform to demonstrate this modeling tool. Okay, so you can go online. You see we made it in 3 languages, so we can add Indian if you think that it's interesting. So how you use it, I will go quick. You just put here your own data in your own situation. So here you describe farming practices, then the fruit fly management, soil management, altitude, season, the presence or not of Docus ciliatus development, wild host plants presence, cucurbit crops in the area, and other crop in the area. So here I randomly selected some decision. We can select one together. So for instance, how much mineral fertilizer do you use on your land? Less than the recommended level, the recommended levels, or more than the recommended level? Which one would you like to input? More? Okay. Don't think about the environment. Okay, so you can see we can answer to each of the questions. So once you have answered all these questions, you can validate your input data, and you made your first simulation. So here you have the output. In the situation that we modeled, we have a very high level of infestation with fruit fly. And here, we have bad farming practices and medium location of land and medium environment. And you can better understand if you develop the tree, and you can understand what's going on in your farm with a level of control of flies. So if you are not happy with that, you can go back to the input and say, okay, more maybe was not a good idea. What does it change if I go to a lower amount of nitrogen, for instance, or what will happen if I have another technique to control the fly? Okay. Now I will show you the second example. So this one is a little bit, so it's only for Frederick, because if you don't like mathematics, it will be a little bit harder. Now it's, when you design a model, after you have to program either in R or in any type of programming language. So this platform was designed in order to provide a free and user-friendly platform to help people design their dynamic models, and also to share the models either with friends or to the world, and also to use them. So I'll go quickly. There are 4 sections. One, proposed tutorial and documentation. One is the list of models that the modelers decided to share with you. There is a modeling forge where you can enter some equations, and here there is a simulation center. I don't have time to go into details, but I can just show you the tutorial and documentation session. We made very nice and simple videos in order to present each of the functionality of the platform. So it's very self-explaining, and if you you can record the address of this modeling platform. You can register. It's simple. You just type your email. You will receive an email and you validate it, and you have access to that. If you want to try, I will be happy to help you connect and have your first step in this. Okay, so I will just show you an example, and after I will conclude. So I go back to Keynote. So this is an example of use of Model Builder to help design some control strategies for aphids on cauliflower. Okay, so this is a field of cauliflower. I think you do have cauliflowers in India, because we had in the restaurant, so it's my guess. And so these are bad guys, the aphids. And this, with a simple model, we were able to design the development of aphid population. So using a very simple ecological model, a Lotka-Volterra model, very famous in ecology, we were able to analyze the effect of a strategy by establishing flower strips in the field of cauliflower, because it will attract ladybugs that will be able to control the aphid population. So here you have in blue the aphid population, and in black you have the ladybug population. So you can see dynamically after day 22 we already control all the aphids in the field. So we can understand that by modeling. We can also test another strategy. For instance, we can say what is happening when I apply two black soap treatments, one here and one there. So you can see the dynamic and the effect of black soap. However, after day 22, we still have a high level of increase. And after, you can make any complexity. You can say, okay, what is going on if I use not only flower strips, but also black soap treatments? So these are the two dynamics that are models. So using that, we can say, okay, if I compare the three strategies, the best one with regard to the efforts that are needed and the impact on it is just to use flower strips. So it's just example. You can make anything you want with a model, with a platform, model builder, which is very generic. So in conclusion, the two platforms that I showed you, IPSIM and Model Builder, are free, they are open, and they are very user-friendly to design models, and they are made to help collaboration. So I hope maybe at the end of the workshop we'll be able to think about modeling project if you're interested. Modeling is not a silver bullet. Come in. It means that you always have to use other approaches such as experiments. However, even if it's not a silver bullet, it's always very useful when you organize and you structure research programs. And it helps building a bridge between scientific disciplines and various types of actors. And I would like to stress the importance of conceptual modeling. For me, it's the most important stage when you design a model. So, Nandri, for your attention. Thank you very much.
Okay. Thank you. So we can have one or two questions, quick questions.
For the talk. I have a question just about the model. Yeah, it's better like this. My question is, in the model that you showed us, there was no answering like unknown answer. Mm-hmm. So when you design the model, do you take these uncertainties that you can have in experiments?
Yeah, it's a good question. When we have this, because it happens some data sets, we don't have all the input variables. In that case, we perform a sensitivity analysis by using all the possible options, and then at the outcome, you have some distribution of the outcome. So if you don't know the answer, the interface that I show does not allow to do that. But when we are conducting a real research project, we can use a distribution for the unknown variables.
Yeah, thank you, Jean-Noël. So at some point you mentioned the fact that one can use conceptual models to help writing white papers. One of the purposes of this workshop maybe to have a white paper as an outcome, and I was wondering whether your platform could be used in that purpose, or was it something else?
Yeah, thank you again for the question. The fact is, IPSIM is actually very close to a conceptual model. The only addition is that we are using a software called DEXI, designed by a colleague in Slovenia. And it's very easy to transform the conceptual model that can be captured very easily under DEXI into a simulation model, a static model. So why not? It can be a way to think about how we can sit around a table and think about common ideas in order to control one pest in a given crop, or more generic, or in a more generic way.
Thank you.
Any question from an Indian colleague? I had a French colleague, but was it easy to follow? Was it very strange? Did you understand anything? Yes? Yeah? Yeah. So it all depends on the choice of the input variables. For both approaches, I don't have time to go deeper into details, but we can translate projections, for instance, in weather, and you can use that as input variable of IPSIM model, and then it will become a predictive model for the future. Usually, IPSIM is more ex post, is more used ex post in order to understand what went on and to provide some guidance in order to optimize crop protection strategy. And for the design of Model Builder, well, it's your choice because Model Builder is just a tool. And if you want to design a predictive model, then you have to choose input variables that will allow you to predict the future. So to my understanding, both approaches can be used either to understand ex post situations or to provide future status of the system being modeled. Yeah, again, it depends on what you do because you are the modeler. But usually for IPSIM, the data that you need, it's what I showed here. It's the, for if you are dealing with pest and pest control, it's usually you need some data on soil and weather. If you are dealing with airborne diseases, often you can skip the soil. It's not that important. For other pests who live in the soil, they can be important. Then cropping practices, for me, it's central because I'm an agronomist, so maybe I'm biased, but there are always very strong effects of cropping practices on pest development. And depending on the pest that you are considering, you might need information at the landscape level. So, but after, again, it's a choice of the modeler. So it's hard to have a generic answer because it depends on the question that you have and the type of model you want to design.
Okay, so in the background, whether it's used linear or some sort of model, I mean, we are just giving the data. In machine learning also, it learns from the data and then it provides the model. So what sort of model does this modeler provide?
Yeah, what I showed does not rely on statistical or artificial intelligence data. You can have that beforehand, and you can decide to use that if you're interested into that, to implement that whether into IPSIM or whether into Model Builder. But so far, I didn't have in mind to implement that type of functionality in Model Builder. Actually, I didn't have time to present, but we are progressing in the functionalities of Model Builder. So at the moment, some colleagues are coding in order to provide parameter estimation, to provide evaluation of the quality of prediction, and to provide the assessment of sensitivity and uncertainty analysis. So 1/4 functionality could be okay, but I think the market is not as big because it's better to use dedicated tools such as R or any type of other tools, quite high-level languages in order to use these innovative techniques.
Okay, thank you. Thank you for your
You're welcome.
Yes, Dr. G. Karthikeyan. 1 minute, Karthik. So before the presentation starts, we have a small a request from all the speakers. Because as per the norms, we go to prepare the proceedings from each presenter. Although we are noting the points, what I discussed with the organizers that it is better to the proceeding comes from the presenters, because you know the better than us what you want to put in the proceedings, because it will speak about the international collaboration of the new projects. I think particularly all the colleagues from the French, you kindly put your points, what kind of collaboration you want from the Indian scientists, like that you keep your proceedings, 2-3 points you give us, so that we will put it in the proceedings. Otherwise, we will prepare our own proceeding, but we want the input from all the speakers. Thank you.
Yeah, thank you. I'm host. Respected chairpersons, my dear friends from India and France, good afternoon, all of you again. So, first of all, let me thank the organizers for giving me the chance to stand here and present my works on begamoviruses. So, I will be presenting, or another 10 or 15 minutes, I will be talking about the begamoviruses and their, something happened. The title is very small, but I made it very big but small. Oh, it's okay. So, begamoviruses with the special reference to cucurbitaceous crops. I'll be like to talk and discuss about their diversity and how the variants are coming up one by one to infect vegetable crops, and also their management strategies, present strategies available, what all the future strategies we need to have in order to contain the spread as well as the control of the disease. Next slide. So here, first, like to give an introduction about the vegetable crops. As you are aware, vegetables are the major source of vitamins, minerals, dietary fibers, antioxidants, etc. And in India, vegetables all put together, which is cultivated in around an area of 12 million hectares with a production of around 220 million tons. India is the second largest producer of vegetables next to China, and the annual losses due to these viral infections. As far as when we talk about the diseases of vegetables, I was wondering, our friend from Ooty did not talk about the virus disease in potato. Of course, they are concentrating on fungal and other bacterial diseases, but the virus diseases, no. The loss is varying from maybe 10 to 100, even 10%, because it totally depends upon the time of infection and the type of the viruses involved in causing the infections. So there are about more than 200 viruses infecting vegetable crops put together from different groups of viruses or the genera of viruses like Begamo, Party, Taspo, Kukumo, Crini, Poliro, Tobamo, Como. Several viruses are infecting. And the problem now is the mixed infections, especially in vegetable crops, that make the thing to very difficult to control the disease. And the reasons are the intensive cultivation. Throughout the year, for example, the villages which are very nearer to our campus, Tondamutur, throughout the year we will have a tomato crop. So the inoculum, not only viruses, all pathogenic inoculum are always present throughout the year. So stage by stage, even single farmer, due to the what do you call the price flexibility, the farmers used to grow or raise the crop in different months. So thereby continuously within his own field, the inoculum goes from one field to the next field, likewise. So throughout the year we will be getting. So that intensive cultivation as well as continuous cropping are the major factors which are determining for the severe occurrence of the diseases, especially the virus as well as the begomoviruses in general. And in regard to the virus history, our plant virus research was 100 years old, more than 100 years old. But viruses are very simple in composition, only the nucleic acid and code protein. Maybe in few viruses will be having envelope. Very simple, but they are available in variety of shapes. And though it is very tiny, but most difficult to control as well as the diagnosis. We experienced with corona itself. So for example, as far as the plant viruses are concerned, during 1939, there was only 89 viruses, total numbers infecting all put together vegetable, horticulture and agricultural crops. But slowly there was gradual increase in the number of viruses, viral species, and now it is more than 200 and 250 as on date, because after the submission of or dumping of data from the NGS, it has already crossed to 2250 species. So that is the status of the plant viruses as far as our cropping system is concerned. So these are all the major group. I have just mentioned few of the viruses. Even with the Begamo, not only tomato leaf curl New Delhi virus, which is one of the predominant virus in Tamil Nadu and also in India, but however, there are several variants from different regions within India that we will be looking that one. And also in okra, both yellow vein as well as Indonesian leaf curl, chili leaf curl, several viruses in cassava mosaic viruses, and similarly in party papaya, ring spot, zucchini yellow mosaic, chili veinal mottle, then in cucumber virus, cucumber mosaic virus, which is one of the dangerous, and tabamo virus, cucumber green mottle mosaic virus, and TMV, tomato mosaic virus, as well as the TASPO viruses, the necrosis symptom causing viruses like TSV as morning we had no TSV on groundnut, cotton, groundnut button necrosis virus, capsicum chlorosis, watermelon button necrosis. They are very severe outbreak we are getting in India. Similarly, the polioviruses. These two groups are emerging now. But though we are talking about the begomoviruses are emerging, but already emerged, but new viruses are also emerging in that group one by one. So as I said, they are all in a variety of shapes. See here, flexuous rod, spherical, or pleomorphic, or bacilli type, or different types, but the begomoviruses are Gemini. They are all twin particles. They look like the bullets. So based on that only, they have coined the name, the geminate, or the Gemini viridae comes under the family, and they are twin particles. Either they are monopartite or bipartite, with the approximate genome size is very simple, 2.7 kb only. But there are about 14 genera under this family, but the begomoviruses alone has about around 70% of the viruses coming under this begomovirus from the Gemini viridae. And associated with the satellite also. I think that Dr. Urbino going to talk about that satellite viruses tomorrow, satellite molecules. And both alpha as well as beta. Delta is also there, but we predominantly getting only alpha and beta satellites. And similarly, they are infecting, there are about 243 begomovirus species infecting only vegetable crops among the horticultural crops. And Bemisia tabaci whitefly transmits the virus in a persistent, circulative, but not propagative manner, not in a propagative manner. So pathogen detection, we need to know what is that. Nowadays, no confusing whether it is due to deficiency or due to some other fungal or bacterial pathogens, because necrosis is also one of the symptoms caused by the viral diseases. So we have several methods. Of course, here for the pathogen detection, the cultivation may not be possible for our viruses, but other serological as well as the molecular methods, either protein-based or the nucleic acid based methods, or the sequencing as well as the biosensors and the non-invasive detection methods are all commonly used for the detection of the viral diseases in the horticultural ecosystem. So I'll be talking about the cucurbitaceous begomoviruses especially. So we have 7 agroclimatic regions in Tamil Nadu. Throughout all the zones, we will be cultivating cucurbitaceous crops, except a few areas like high rainfall area and also some of the other hilly areas also we have the chow chow and all, or chayote. But however, in all the places, we will be recording the begomoviruses and the characterization. So you see first the symptoms. When you talk about variety of symptoms are producing. So you can just for to know the crop, familiarize with the crop, I have included the fruits also. So the bitter gourd, they produce different symptoms. The leaf curl, yellow mosaic or ordinary mosaic or mosaic mottling, and similarly on pumpkin also you can see kind of chlorotic spots or chlorosis as well as the mosaic mottling look like yellow mosaic, and on the ridge gourd also you can see yellow mosaic and the yellow mosaic, see here yellow mosaic on ridge gourd, leaf curl, puckering also could be seen sometimes. Then on the ash gourd, we will be getting mosaic, yellow mosaic as well as the yellow mosaic. And on the bottle gourd also you will be getting mosaic, a typical mosaic mottling and the filiformity of the leaves, or sometimes they become narrow, leaves become narrow. And on the ivy gourd also you will be getting yellow mosaic, and snake gourd, typical characteristic mosaic mottling can be seen. And on cucumber, there also yellow mosaic will be common symptoms. And muskmelon also, yellow mosaic is the predominant, and the leaf malformation or deformation of the fruits also could be seen in majority of the crops. And similarly in chayote also, typical mosaic mottling as well as the puckering of the leaves are the additional symptoms. This is a severe outbreak with yellowing or yellow predominancy on the leaf surfaces. So normally these begomoviruses are characterized, diagnosis is easy, but they are characterized by a special method, by the amplification of the genome by RCA, rolling circle amplification. It is a simple isothermal reaction by using the special enzyme called 529 that will able to produce concatenameric DNA. So from that, by restricting those sequences, we will be getting the 2.7 kb genome of this viral sequence. So that is highly useful for the diagnosis as well as the comparison of the viruses. So this is one of the based one general group they formed the viruses, which the begomoviruses which are infecting the vegetable crops, mixed infection, are only on solanaceous crops, and solanaceous and malvaceae crops, solanaceae, cucurbitaceae vegetable crops, and legumoviruses and all, they have group. But however, they are either monopartite or bipartite, and also the involvement of the satellite, either alpha satellite as well as beta satellite. Sometimes both the satellites are also available. So as far as our Tamil Nadu state scenario is concerned, when we have collected the sample from different, by covering all the 7 agroclimatic zones, our analysis by RC analysis, which revealed the presence of different viruses. So for example, these are all the restriction enzymes that yielded different viruses. For example, Coccinia mosaic Virudhunagar virus, tomato leaf curl New Delhi virus, bitter gourd yellow mosaic virus, tomato leaf curl Virudhunagar alpha satellite, tomato leaf curl New Delhi alpha satellite, and also Cinderella yellow vein virus satellites. So satellites as well as the whole genome, viral genome were obtained by this restriction. Normally, the satellites are 1.3 kb only approximately. So both the sequences we will be getting and characterizing the whole genome. And in such a case, in addition to those variants of tomato leaf curl viruses, we could get some of the special viruses also, like squash leaf curl Philippines virus. So we have collected one of the sample from pumpkin from the Udumalpet area of Coimbatore district with a typical mosaic mottling and leaf deformation or malformation with yellowing. Stunting is one of the common symptoms in majority of the begomovirus infected plants. And the incidence was found to be 97%. Then imagine how much the pressure of this virus is present, especially the begomoviruses. And we could get the 2.7 kb by these two restriction enzymes. And it is a bipartite genome with A and B, approximately 2.7 kb. There are slight variations. But however, this DNA A is aligning with the squash leaf curl Philippines virus, whereas DNA B is aligning with the squash leaf curl China virus. So there are variations, there are recombinations, and that we have reported as a new viral species infecting Cucurbitaceous crops. So here you can see this is the alignment. So either they will not be aligning with, not aligning with the tomato leaf curl New Delhi virus, and which will be aligning with this one only, DNA A with the Philippines virus. And similarly, DNA B is with the China virus. So likewise, there are several variations among the begomoviruses which are infecting the Cucurbitaceous crops. Then again in coccinia, from Dindivanam, there is a Viluppuram area of Tamil Nadu, we have collected some of the samples which are showing different kinds of severe deformation of the fruits as well as the leaves, and that resulted in a begomovirus amplification, begomovirus genome amplification, and we got both DNA A and DNA B. But very interestingly, that DNA was aligned with the Lufa yellow mosaic virus with 78% only. As we know, the demarcation level for the species level, demarcation level is 91% homology for begomoviruses. And similarly, DNA B had 76% identity with the tomato leaf curl New Delhi virus only, but it is not Indian isolate, it is with the Pakistan isolate. So likewise, no, there are several variations among them. And from Chayot also, from Nilgiris, as madam was telling, Kavita madam, and there also we have collected some of the samples showing characteristic mosaic mottling symptom, and we could get only DNA A. So we are under the process whether it is monopartite or bipartite, but however, we need to check again, no, bipartite only, but we didn't get B. But this DNA A was aligning with just at the 91%, that is the demarcation level, 91% with the tomato leaf curl New Delhi virus isolate of China, India, Spain, India, as well as the Pakistan. And we could not able to get to that one, the DNA B, but however, that is at the marginal level, at the different level only. And when you talk about tomato viruses, there are also in tomato also, there are several begomoviruses as we are aware. Again, the tomato leaf curl New Delhi virus is infecting, and there are also several variants nowadays we are getting up. And the major symptoms are curling, puckering, yellow mottling, shortening of the internodes, and sometimes we will be getting 100% yield loss due to severe outbreak of the disease. That too which coincide with the infection of peanut buttonacrosis virus, that total plant will be collapsed. In Tamil Nadu, 73% of the tomatoes are produced from these four districts, Dharmaburi, Krishnagiri, Salem and Dindigul, and the symptoms are varying, starting from leaf curl, but we don't get yellowing symptom here. Only leaf curl, stunting, malformation of the leaves, even sometimes fruits also, but we will be getting some of the weeds also get infected with the same viruses. In the ecosystem of tomato, we have collected several weed samples, and that also turned positive for the viruses which are acting as the
5 more minutes.
Okay, sir. So collateral host for the viruses. So here, see, at that time, you know, when I was starting my research on viruses, maybe 15 years back, we could get only tomato leaf curl New Delhi virus. But nowadays, even in Tamil Nadu itself, majority of the genomes are aligning with the Bangalore virus only. But in this, particularly in these districts, we have randomly collected and analyzed, we are getting different viruses, not only New Delhi, and also Palampur, Bangalore, Gujarat, Kerala, and also satellite also from different categories, Bangalore, Gujarat, and also Multan, cotton leaf curl Multan alpha satellite was also present. That's what I asked that question. The cotton leaf curl virus is not present in Tamil Nadu, but the satellite is present, alpha satellite. And similarly, the beta satellite of tomato leaf curl New Delhi virus is also obtained in the tomato sample. So this is the scenario. So all are within these groups, one or two as a mixed infections, in some cases single infection, or in some cases with other viruses, not Begoma viruses, with other viruses like CMV sometimes, or cucumber mosaic virus, or with peanut butter necrosis virus, also occurring as a combined infection. That is a major menace nowadays, the combined infection. So this is the phylogenetic tree constructed using the DNA A of tomato leaf curl, New Delhi virus, Palampur virus, Bangalore virus, as well as the Gujarat virus. They are all formed different clades. Separate clusters are formed with them. And similarly, see, DNA B also, they are forming a distinct group, and the alpha satellite also, they are aligning with the concerned group of alpha satellite, whether they are tomato leaf curl or Bangalore, New Delhi or Bangalore, like that. So this is the scenario again. So this is the Gujarat virus, as well as the Gujarat beta satellite also we have characterized. And recombination analysis also we did, and some of the viruses between the isolates from one crop to the another crop, like tomato with the cucurbitaceous isolate, they have recombinations, either in the DNA A or in the DNA B. A small portion were identified in the recombination analysis also. And with this background, we have developed the infectious clones for the cucurbitaceous Begoma, one of the cucurbitaceous tomato leaf curl New Delhi virus. And we have developed the agroinfectious constructs, and that agro clones were inoculated on different, in order to know the host range, on different cucurbitaceous grass, including the tomato. We could get nearly 10% infection in some of the cases, like ash gourd and also squash. And on tomato also we could be able to get more than 90% of the infection by this infectious clone. So and also on wheat plants also we could be able to, but when you add, in addition to DNA A and DNA B, when we include beta satellite, the infection rate was found to be high. Really low when they are inoculated without the beta satellite of these viruses. So these are all the symptom pattern on the inoculated, agro-inoculated cucurbitaceous various cucurbitaceous crop plants, and the titer was also determined. Titer was also found to be very high in the infected plants. And similarly in the beans also we could get both in the French bean as well as in the field beans, we could get this Begoma virus, Dolichos yellow mosaic virus, and that was also aligning with some Washington Dolichos yellow mosaic virus and other Indian strains or isolates from Kanpur and all. That is also distinct. And there are similarly now in the brinjal also there is a complex of Begoma viruses are infecting with a predominant occurrence of tomato leaf curl New Delhi virus and also the Bangalore virus. And chili also. In chili mostly we will be getting chili leaf curl virus, and in addition to that we will be getting the tomato leaf curl New Delhi virus also on chilies. And also in Bendi also both okra yellow vein mosaic virus as well as the Venetian leaf curl also we will be getting nowadays. Of course, nowadays that yellow vein is reduced incidence, but however the Venetian leaf curl was found to be severe. And seed transmission of these Begoma viruses on cucurbitaceous crops was also proved, and that resulted in a transmission of around 12%. So for example, in one of the hybrid, it was found to be 12% seed transmission, and the another one, it was found to be 5%. This is one of the next problem, for the outbreak and the severity of the diseases. And similarly, other cucurbitaceous ridge gourd also, we could be able to get 18% seed transmission in one of the lot, but however, in one of the other hybrid, we could not get any seed transmission. That was proved. And the major menace, next problem is the mixed infection. Within the cucurbitaceous crops, see here, these are all the different combinations of the viruses get in the natural field conditions, but in all the combinations, we will be getting the Begoma. So without Begoma, there is no combined infection as far as the cucurbitaceous viruses are concerned. So that is the scenarios. Then we can imagine how much the Begoma incidence is occurring under the field conditions. Similarly, in the whitefly also, like that of the virus, Begoma virus, whitefly also there are different variants, and there are called as biotypes, cryptic species. That also we could be able to identify. So for example, in the cucurbitaceous ecosystem, that correlated with the incidence also. For example, in cucurbitaceous ecosystem, mostly we will be getting that ACR2 5 group of cryptic species of whiteflies, Bemisia tabaci. And wherever we are getting this 2.5, Asia 2.5, there the incidence was found to be high when compared to the other Asia 1 or the other cryptic species. So this is the grouping, and here also you can see this is the 2.5. It's a very, very big range when compared to the other types. And in the cassava ecosystem, we could get, especially in Salem and the Namakkal and other major growing areas, we will be getting only one group, Asia 1.5, not other group. And similarly, that's and regarding the management, we have good options, integrated management, starting from the resistant varieties. Dr. Andy Kumar was talking about the microbiome, the biologicals, the biological control, barrier crop, and other cultural practices are there. That also we can follow for the management. And the recent technologies like genome editing or double-stranded RNA application, exogenous as a non-transgenic approach. That is also we are working. We have developed the dsRNA strategy for tomato leaf curl nudity virus, and also we have come out with the another one virus, which is also Begoma, but infecting the legumes. For that also we have developed the dsRNA formulation with nano-based formulation also we have come up, and that found to be highly effective under the field conditions. But however, we need to give up approval and other field studies we need to conduct. So summary regarding my presentation, as we see from the presentation that the frequent presence of or the occurrence of mixed infection is the major problem as far as the cucurbitaceous ecosystem is concerned, even other vegetables also. So the recombination between strains and our species playing a critical role, and vector pressure and their environmental factors as well as their cryptic species also play a major role, and the weed host, the reservoir plants available in the concerned vegetable ecosystem, that also play a major role for the survival and the spread of the disease. And their future strategies, though we have quarantine, but there is no strict quarantine, I can say, in the closed wall, because how this Philippines virus has come into India. Likewise, there are several viruses which were introduced from other countries like Pakistan and other neighboring Bangladesh and all. That also we need to take care. And similarly, the climate resilient crops adopting new disease pattern, and the AI should be exploited for diagnosis, disease forewarning, and spot delivery of inputs, smart surveillance and modeling and all they were talking from the morning. Then policy support is very, very important, and the circular economy, and finally, by combining artificial intelligence, genetic engineering, nanotechnology, biocontrol, and climate smart practices, modern agriculture can achieve resilient, high yielding, and sustainable vegetable production systems in the globe. These are all some of the papers we have published from good journals, and I need to acknowledge my sponsors, DBT, SERB, ANRF, as well as Science and Technology, Department of Science and Technology of Government of India, and the USAID, Virginia Tech, Washington State University, and also DSMZ for supplying the, especially for the diagnostic purpose, they used to supply antibodies. Then these are all my team, my boss at ES were Naidu Rayapati when I was doing my postdoc. Then Muniyappan, he was the director of IPM CRSP, and Dr. Nagendran, scientist, tomorrow he will be attending, and my students as well as my research fellows from our laboratory. Thank you very much for your patience. Thank you.
I think you have taken the time of the questions.
Okay, sorry. Yes.
But still, you can encourage one or two questions, quick questions.
Hi, thank you for the presentation. I had a question regarding the double-stranded RNA spray method. So what part of the viral genome do you use?
No, we tried with the two, coat protein as well as the replication initiator protein of begomovirus, especially from tomato leaf curl nudity virus. And among these two, coat protein was found to be efficient when compared to the replicase.
And connected to that, how do you synthesize double-stranded RNA?
No, initially we used, now we are using the commercial kits only. So we are working on the other side also.
Okay.
For this large scale synthesis of the dsRNA.
Okay, thank you.
Thank you. Yes.
Yes,
Yeah, please.
Just a short question. I wanted to know if you have, because top New Delhi was detected a long time ago here in India. So do you have resistant varieties for top New Delhi virus?
For New Delhi?
Yes.
No, madam. Major tolerant we have. We don't have total resistant varieties against that. We don't have resistant sources.
Okay.
Even the wild species are susceptible.
Okay.
That's why we cannot exploit that one. Yeah.
Yes, I think if you can have interaction with Dr. Karthi during free time. We can invite Dr. M. Govindraj for improving the disease resistance horticulture crop, biofortified crops. Yeah, now it's preponed for it.
Oh, thank you.
I think Dr. Govindraj, if you stick on time, we would like to have more interaction.
Good afternoon. Is audible? Thank you. Good afternoon, and respected chair and co-chair and audience and online participant. So I would like to give a very clear presentation so that we can avoid any burning questions. But respecting the rushing time, so I would like to give the brief about I'm taking the different angle of the seminar to contribute how not just the plant and pathogen in interaction, and how the product is being taken to the market with all this upstream research to the downstream product delivery. So with that, I would like to happily present the improving disease resistance in biofortified crop varieties. So basically, all fortification and biofortification is the two sides of the coin. The purpose is the same. We are addressing the malnutrition or hidden hunger, but the approach is two different. So one is a short term, other one is a long term. The biofortification is a long term as we introduce the genetics that repeat your nutrient generation after generation, just like your disease spectrum. So the biofortified crops are mainly is not in the different cultivation practice. It is also similar to any other crop cultivations. So Harvest Plus is one of the pioneer among the global biofortification initiative. It's run through almost 20 years of the challenging program across the globe, including 150 partners and working on the 40 plus countries. So with that, so how our story is begun, so the group can benefit and student can be more, a tangible research can be done in the futures. So the priority comes, the nutrition is, as you know, like pathogen list, there is a nutrition list as well. So we target only those 3 nutrients which cause, among the top 10 the death cases as per the World Health Organizations. One is the iron, other one is the zinc, and third one is provitamin A. Well, somebody can ask why not protein? Protein is generally considered. So biofortification is started with the biofortification priority index as we talk about the model today. So those model is a conceptual model is already taken care of 20 years back in some of the program like biofortification, which include the sub-index to make the major index. What is the sub-index? Is the crop production, is the per capita consumption of the particular crop, stable food, and particular nutrient deficient in the particular geography area. So if you combine all together is so-called biofortification priority index, one can go to the website, Harvest Plus website. You can see the interactive map already built. So this is based on the 13 stable crops, so it's more than 120 countries, which gives the donor, the investor, which gives the idea for the researcher where, which crop and how to invest or do the research. So therefore, I given one example, a selected pearl millet that is highly prioritized for iron content, where the countries as is highly mapped is for India and sub-Saharan Africa. So the next question come, okay, how much nutrient we are biofortifying? this is that one can assume that biofortification not just adding the nutrient into the crop product, it is a regular consumption of those products, okay, a month together or years together, as a part of your stable food, that should give a measurable health indicators. So therefore, the biofortification definition is fulfilled. So please correct the definition. It's not just increasing the bioavailable in edible parts of the plant. It's not stopped there. It should be after consumption, an individual should has a reflect as a health indicators. So therefore, this is that slide which indicate how much the daily nutrient of particular crop in a given per capita consumption should be met. For iron crops, you can keep 80% of your nutrition, as particularly iron, can be fulfilled with these biofortified iron crops, whereas the zinc crops, you can up to 70%. Whereas the provitamin A, you will have much higher, is almost 100%. So this is that based on the nutritional evidences created by our nutritionist. So therefore, the farmers grown varieties, you can see the light green, what you can expect the farmer grown varieties 20 years before. And what has been fixed target, the middle bar, is a breeder has to achieve with all the resistance, with all the challenges of the production, has to reach this level of nutrient in a given country with that program. And the green ones, what as a network partnership, the product finally is achieved that level. So 20 years back, this is impossible, as we are discussing now, is the modeling as the One Health. And this is a very critical example to set, is everything possible if you unite hands and as well as the optimize and use efficiency of the data. So this is the data-driven approach, which gives you that any of the target crops is mostly everything is 100% achieved in 20 years, not single organization, as I said, 40 plus countries and 150 plus partners. And many donors is asked where the demand for nutrition in agriculture. If you think the nutrition is come from always other than agriculture. But you see, the staple food we eat every day, even your lunch, the staple food is our maximum quantity of your plate. So that has to provide the adequate nutrition. Therefore, that is not being done because farmers not going to demand nutrition to grow his own variety or her own variety. So if it is a high deficiency rate, it should be the first scenario where we call it as both public and private sector. One can develop the product, all type of the biotic and abiotic stresses, so-called target product profile, TPP. You must be heard, many of people from ICAR or private sector. That's the blueprint of a variety to be developed in next 5 years. This is the evolving mechanism. So one has to build all the needy mandated traits into the one product portfolio. That's the critical. So therefore, we are doing advocate with many of these donors as well as the research priority and policy setting background. And this should be taken very enough care. It's where is the high deficiency. Say for example, India is 50% of the woman is anemic. The question is how supplement going to support in a long term, because agriculture should come to the picture. So therefore, the product profile should have those crops which where is feasible. It should have those traits along with the disease resistance. So I come to the central topic of the seminar. The crop development is taking care of the entire chain of process. It's starting of the discovery, in the morning set and even the afternoon we are discussing a lot of discovery, upstream research that has to be evaluated in the product portfolio and integration of the product pipeline. So which is not exceptional than the regular or traditional plant breeding. In the same way, what we do, we act as we include as many as discipline as possible in this case, not pathologists, not entomologists alone, not even statistical alone. We also include policymakers, the socioeconomists, we also include the nutritionist. So those two, a champion is going to decide what to breed, what not to breed. Because the breeding of the product is for consumer, not just for farmers. So farmers will focus productivity, but consumer will prefer something else, the quality of the product. So therefore, it is very critical to intervene any of the new traits, just like what disease resistant in nutrition, to combine together is a very challenging job. But however, the team has been achieved so far. The goal is not to trade off anything, which is farmer preferred yield or disease resistant, or consumer preferred color or taste. This is a very critical example. That's what I would like to present. So that how we screen is just like any other program. This is one of the example slides I used from ICRISAT when I was working with the team for pearl millet. So, every segregating generation, so after crossing, we used to screen a lot of germplasm and crossing segregating material. So you see almost 40 plus entry out of 90. So almost 50% is qualified, is more than, almost all the pathotype for downy mildew resistant. So the downy mildew is the major devastating disease in pearl millet. Therefore, that is a part of mandatory screening, including the high nutrient dense lines. So this bar indicates how the 5 different pathotypes, it's shown the resistant. You can see all the biofortified lines are given adequate resistance. So what I mean resistant is mostly tolerance. So this is also done. It is highly possible in downy mildew because it's a 50 years work. People are doing both conventional and molecular mechanism to build the resistant gene, pyrimidine. But when it comes to blast, that is not possible. It's more, is evolving very faster than DM, and also is the very latest disease. To control, and we need to have the hotspot location to screen adequate screening. Therefore, the data also reflect all the entries what we have screened, we have less number of resistant. But the same case for any other breeding program is the random data. So similarly, we also use, like in a genomic data, how we can avoid the G by interaction because we can't do enough phenotyping with the available resources. So we do identify some of the QTL or SNP markers for blast. You can see the intracrust line, male and female, and also susceptible line. But we also work, is developing diagnostic markers, which is part of, daily business of the breeding program in any hybrid crops. So these are the diagnostic markers, which include both disease resistant, high iron, and high zinc. So you can have the packages. So that set of traits should be screened all the 1,000 lines in a season. So you will select. So this is the efficiency. So then the question will be asked, are you doing in one location as the disease is spread every 200 kilometers is differently behave as the environment is varies. So you can see the India's almost all the millet grown region and states has been evaluated. And the data clearly shows that there is an integration of both stress, biotic and abiotic. Millet is always in a marginalized environment. But in the nutrient, on top of it, both resistant, both blast as well as downy mildew, this is highly possible when you have the multi-location data. So therefore, we would like to always thank the partnership what we had with the private sector as well as the NORDS team, including all state agriculture university, RICAR institution, who provide, as a voluntary as well as sometimes is a project-based location to test. Otherwise, it is not impossible. Therefore, multi-location testing is very important to take the next level of your upstream research to make the downstream product. And in another case is the beans. So biofortifications work on most 8 to 10 crops. So we are looking after both, all the global south is 40 plus country. And this is one of the data from Kenya, is the common bean is in any area we call it as mostly the rajma. So this is very, very interesting story again, just like pearl millet, which is high in iron. All the biofortified lines, you can see the level of iron level, which is also shown is a very good multiple, double or triple disease resistant, which has been selected for field condition. Here there is no genomic intervention. What I'm trying to say is that as long as you visibly or consciously select either disease resistant or nutrient dense, is also going to be in your seed. As I said, it's all about playing with the genetics. So this all we need is the new technology to reduce the cycle. There comes the modeling expert should help us what number of cycle we are going to make more efficient use of resources and also efficient of delivery of the product. So the final one is that we do have the good resistance in a biofortified crop culture. So it is not to worry about. Some of the literature says all the biofortification will maybe trade off the yield or disease susceptible. I don't see any evidences so far. Maybe this house has a lot of pathology experts can bring some of the novel topic to discuss further. So another one example story I would like to highlight here in this seminar, the rust-resistant zinc wheat released in Pakistan. You know, Pakistan is also one of the largest wheat growing country and wheat consuming country. And Sindhu region is occupy almost 50% of the wheat area in Pakistan. And this is the one single variety occupy 40% of the area after 3 years after release. This is the biofortified zinc variety, but that is again dangerous. One variety is occupy a larger area. But how do you resist? So we are working on the pipeline aspect. But what I mean to say is that it's almost 40 ppm zinc. It's the number one in Pakistan, and in the level of zinc, okay, it's almost 50 to 60% higher micronutrient. It is also resistant to yellow rust. It is also tolerate heat tolerance. So you can harvest. I think a couple of years back, the heat wave is very big in India as well as in Pakistan. The wheat yield is drastically go down, and this is those farmers who grow these varieties had a very happy harvest. So this is very important variety. This is the success story of is combining the most demanding varieties and traits together. And this is the seed production, and India is one of the best example. And as I said, Pakistan, here also we did for zinc wheat and zinc rice and pearl millet, which also largely produced. And this has been consumed by millions of farmers and consumers. And mostly farmers, when I say only on farm, who grow, who eat, not buy from the market. So those calculation is yet to be calculated how much is. Another thing is one of our project in Indonesia. So this is that, and again the disease resistant, you can see the UPP either 6 and 7 and as well as 4, which has been identified for high zinc rice in Indonesia. It's going to be released at the end of this November at the Ministry of Agriculture. And this is that is combining with multiple disease resistant as well as high zinc level. So these are all the partnership based breeding. And what I'm trying to say is that the selection makes a difference in terms of any of the product driven approaches or the project. But what we have done mostly we given the freedom to the partners who being expert, but the product advancement is always happening with the multiple discipline involvement. So this is all expertise should be there to advance. So otherwise we may be biased in selection and advancement of some certain product or certain trait biasness. So this is the better example. In Indonesia also we are following the rice crop. To conclude, as I said, nutrition, how is the highly heritable and transmission one generation to other generation, just like your disease resistant. This is highly possible to combine both traits in a single variety or hybrid. So therefore, this is highly recommended to go ahead. There is no stigma of any ignorance of the trade-off, which has been highlighted, but that's maybe the random. We need to look at the very broad spectrum and large scale value. The second one is the most integration of understanding plant nutrition and how it is really the interaction with physiology, because environment is very critical. So those has to be focused, very host plant resistant aspect. And finally, all we know as both micronutrient and macronutrient play a lot of role in plant nutrition, and we need to understand that's also contribute to the control of plant disease. Certain example, say for example, phytate is although anti-nutrient, it is also involved in the defense mechanism in the plant health. So one has to look at, if I want to increase the bioavailability of good nutrition, I need to reduce the molar ratio of the zinc and phytate. One should play very smartly, okay? So otherwise we are doing injustice to the plant health system. So therefore it is very important, because most of the phosphorus in cereals stored as a phytate. So it's 80% of the phosphorus stored in cereals is a phytate. So we can't reduce to the below optimal level. So one should understand the crop optimal level. So therefore, such understanding of the genetics and mechanisms is very critical. The challenge is always with the environment. As you said, every 2 kilometers evolve the pathogens, and also the environment is most, especially the soil environment, is very critical. So where we come to that next phase of biofortification 2.0 is the microbiome, is agronomy-based biofortification. Is the genetics one that contribute 50%, but remaining 50% contribute to the input-based agriculture. Either it should be yield or should be the nutrient or disease resistant. So therefore, we need to combine the gains come from both management as well as genetics. So this is very critical area one should focus. And next one is the barriers, always that environmental is very critical, but we have now tool modeling and genomic tools are available, and that is we are far better we can handle those challenges. And finally, the limited germplasm is screened. I cannot say is available. It's limited germplasm is so far screened is to identify the multiple traits which has either should be in the germplasm unit or should be the breeding population. One should to critically look for it and select and for their either upstream discovery research either at the student level or the project level. This should be is more cautious and one should characterize fully. Finally, what we recommend as part of this conference, as I said to the chairman, you can also write and indicate what to be presented in the proceeding. I think I would ask that it is very important to focus the association between nutrition and disease epidemics. Is it true or not? Is that any expectation if the more nutrition plant will give more nutrition to the pathogen growth or infection? That area one has to explore. That is very critical part. And the second one is the integration of biofortification with the durable resistant. Okay, when I say durable, the pathogen as fast as evolve, we need to evolve as the pipeline of the varieties. With the expert like you, it has to be very critical for adaptation of those new innovative varieties at the farmer field. There we can contribute food system. Then you talk about security, then you talk about One Health. I think this is all I would like to share. If any more question, I would like to happy to answer. Thank you.
Thank you, Dr. Govindaraj, for his excellent presentation. So I think if you have a few questions, quick questions, we can interact.
Sir, nice presentation. Nice presentation, sir. Previously, very long back, golden rice was developed in India, particularly Tamil Nadu. Is it the same as like biofortified species, a product?
Okay, you are asking the approach what we followed in biofortification, am I right?
Yeah.
The method. This is mostly the conventional. So far released, we have 450 varieties in 40 plus countries released, all based on simple hybridization, which follows the traditional plant breeding method, conventional breeding. But having said that, we are not against the other approaches to develop biofortified crops. So that's where we are moving to other technology where we can fast and speed up the development of biofortified varieties, which has more other traits preference. So we need to move with the new technology as well. So the golden rice now is being tested in Philippines. So we are also engaging in that review committee, and this will be public in a couple of years from now. Thank you. Yes, please.
So suppose the soil is very poor nutrient status, micronutrients. How do your biofortified varieties perform? Is it depend on the soil nutritional status, the performance of biofortified varieties, or it will work in a very poor soil where micronutrient is already depleted?
It is very good question, but the purpose of the biofortification not discriminate the poor geography or low fertility or high fertility. The purpose of the biofortification to serve the better environment, better endowed environment. That means your optimum nutrition, everything is there. But we also have some data which tested with our curiosity, how does this low zinc or high zinc wheat perform in low P soil or is the low fertility soil? And in fact, iron millet, how low P in West and Central Africa. So this all is doing good, but we will not say this is only can do the best. But the nutrition level is our focus. Are there any reduction in those nutrition in those poor fertility soil? But when you grow with other variety, this always be the top. So that what I mean to say, the nutrition level still higher than the other varieties. So therefore, we are not in deep dive into those research. So the purpose is this, then we stopped at this level.
Actually, the high nutrition, whatever you are accumulating in the grains, is due to its better absorption capability.
Absorption, and it's not always. That's what I'm saying. It is also retranslocation. As the is number of grains what you have. The more drought in crop like pearl millet, you will have more nutrition because the number of grains is affected, the reproductive stage. The less grain, you have more nutrition harvest. So, but otherwise, saying in general, if your optimum yield is there, this is not going to reduce your nutrition level.
Yeah, but still, it's worthwhile to correlate these two things. So
Yeah, of course.
Poor
One should
nutrition.
aware, yes, yeah. Thank you. Yeah. Very much, you can mention all 8 crops, you have plenty of sources now in elite background. Okay. So you're ready to use, yes.
Okay. Thank you, Dr. Govindraj, for excellent presentation. Now let us welcome Dr. Sisa on how vector transmission participate to dissemination of the beta satellites in the Bergamo virus tested areas, infested areas. I think same time for you also, 20 minutes.
I thought I would have a larger time. Okay, so I'm going to speak about Bergamo virus disease. And as it was just introduced by my colleague, I will not spend much time on the virus, but I'm most interested on beta satellites. So I'm working in Montpellier, so no, just to show you, but I don't know which one I have. This one maybe, yes. So I'm working in the south of France, not far from Mediterranean Sea. And this is our building at Montpellier. And I'm working in a French unit, which is Plant Health Institute of Montpellier. And in this institute, I'm working in a team which name is Characterization and Modeling of Epidemics. And we are working on different we are studying different steps of epidemics, from the evolutionary of viruses until the sources of viruses, dynamics, until the evolutionary consequence of management strategies. So in this team, we have 3 main models. So the one is Charka virus on Prunus, which is a quarantine virus in France. We are also working on viruses on cereal, which are very important, and the vectors now cannot be controlled because the pesticides are forbidden. So there is a real challenge to control these viruses. And then the one that I prefer, the tomato yellow leaf curl virus on tomato, which is present in all the Mediterranean basin. So, as you know now, Bergamo viruses are very important. They affect a lot of type of crops, and from cassava, cotton, and vegetable crops. And they were detected around the 80s, and they are transmitted by the whitefly Bemisia tabaci, which is present in all the intertropical area, and they are encapsulated in Gemini particles. And they can have monopartite or bipartite genomes. And so this is the geographical dispersion of these diseases. And at the end of the 90s, some scientists discovered that some viruses could be that they found were not, they could not reproduce the disease because they lack a component. And then it was mainly on cotton disease that they found that this disease could not, could be reproduced, could be inoculated to plants only if the virus was associated with a molecule which is called a satellite. So a satellite, it's a mobile genetic element deficient for the genes of replication and encapsulation. So it's not a virus, it doesn't have any homogeny with the virus. So it's something which cannot be, which is not independent. It needs a helper virus to be replicated and be transmitted. So it's like a parasite of a virus. It parasites the function of the virus to be replicated in the plant, accumulate, and be transmitted. It encapsulates in the same capsid protein of the virus, not with the virus, but in a dependent capsid, and then it is transmitted with the virus by the same whiteflies. And its genome is composed of only one gene, which is beta C1 gene, which is the determinant of function of these molecules. Begomoviruses have 3 different types of satellites, but the most important one is the beta satellite, which has this gene, because it increases the symptoms of the disease, the viral disease. And it is just this gene which is responsible of this increase of the symptoms. And also, it can increase the accumulation of the virus because it suppresses the plant defense system and then allows a higher accumulation of the virus. And so the dispersion of the geographical distribution of these molecules are mainly in West Africa, Asia, until east part of Asia. And there is no satellites in the Mediterranean basin except in Egypt and now in Israel and Jordan. And there was arrival of beta satellite in the US with cotton, but then it was eradicated. So I don't know why the slides are, it's because it's a PDF, excuse me. So the beta satellites, there are 2 types of association with viruses. In some, as I told you, like a malvaceous plant, like cotton, the satellites must be there for the virus to infect the plants because the virus cannot go over the defense system, so it needs the satellites to infect. So in this time, the satellite is like a viral component. It's not a viral component, but it acts as a viral component because the virus needs the satellite. But for the other viruses, begomovirus, there is no need of this satellite. But they can replicate the satellite. So they replicate the satellite even if they don't need it. And sometimes it increases the symptoms of the virus. And then it creates a new disease, by fact. And so you have this, I'm sorry because the slides are not well positioned. You have the beginning of the slides. Okay, so there is 2 types. So you have these mandatory associations which maintain the satellites because the satellite has to be transmitted for the virus to infect. So in cotton, for example, you always have the satellites. If the virus is there, the satellite is there. So this system maintains the satellites. But in tomato, for example, with tomato leaf curl New Delhi virus, for example, you don't need the satellite. The virus can infect by itself. So you have these 2 systems, and this raises some question about how can this satellite be maintained when the virus doesn't need it. And if you see in the literature, there is no specificity between the virus and the satellite, So we have now around 400 begomovirus different and 100 beta satellites, and maybe all of them can be replicated by all. So this makes a very huge amount of association, which can be dangerous. So this is a very big problem for Mediterranean area, where you have a lot of vegetable crops. We have 6 different begomoviruses affecting mainly tomato and cucurbits, and we are very afraid of the introduction of satellites there. And in 2012, yes, thank you. In 2012, a beta satellite was detected in whiteflies in Jordan, but the disease emerged only in 2016. And we don't know what happens between this time. And when the disease emerged, the scientists there, they sequenced the beta satellite and say, oh, we have a beta satellite. They look in the literature and found that this beta satellite comes from Israel. And they said, but we didn't have it. But it was published by someone else who took some samples and found the beta satellite in 2012. But the disease appeared only in 2016. So now we are really afraid because, so it's there, it's in West Africa, and we think that it will arrive in the Mediterranean basin because of movement of whiteflies, intensification of vegetable crops. So what we are studying at Montpellier now is we have begun some studies to prevent this emergence of new disease, and we are studying the capacity of maintenance of beta satellite by a virus which doesn't need the satellites, because the virus we have here, these viruses, they don't need beta satellites. So we have studied the capacity of maintenance of beta satellites by these viruses, the dynamic of accumulation in planta, the cost for the virus, and the transfer of beta satellite between hosts and vectors. So this is the first thing that we have done to see how they can be maintained by our viruses in the Mediterranean basin. So we use agroinoculation methods of clones, and we inoculate beta satellites isolated from okra in West Africa, which is cotton leaf curl Jezera. It's an error, beta satellite, not a V, but a beta satellite. And then we inoculated tomato plants and follow the infection rates and accumulation, DNA accumulation. And we found that with the virus, we know that we have these type of symptoms with our TYLCV, tomato yellow leaf curl, and tomato yellow leaf curl Sardinia virus. But when you add the satellites, then you have very important symptoms. You can see here the size of the leaves between the healthy plant, the virus, and the virus plus the satellite. So we have 100% transmission of the satellite with the virus, 90 and 50. So it depends on the virus. And then we did it also with Talk New Delhi virus, because Talk New Delhi virus was introduced in Europe in 2012. And it's the strain which was introduced in Europe could infect only cucurbits, not very well tomato. And so we tested how this satellite could work with tomato leaf curl New Delhi, and then we did the same experiment. And then here you have the disease of you have the healthy plant, the plants with the virus, melon and zucchini and melon. And when you add the satellite, you have very severe symptoms. And then you have also here the size of the leaf. So this beta satellite increase our symptoms, viral symptoms. Then we did the same thing with another satellite that we found in Iran, which was isolated from a papaya plant co-infected with TYLCV. So we did the same thing with our virus, and then we saw that this beta satellite could be replicated by the tomato virus, but not very well with TYLCV Israel strain, but not with these two viruses. And it could not be replicated by tomato leaf curl New Delhi. So you see, it depends on the virus, it depends on the host. and it depends on the satellite. So you never know what will happen. Then we follow the dynamic of accumulation of satellite with our virus, which is tomato yellow leaf curl virus. And then we found that this is the dynamic of the virus alone in the plant. And when you add the satellite, so you see that the virus accumulates at a lower level when the satellite is there. So there is a cost for the virus. And the satellite began at 11 days post-inoculation, and from 18 days post-inoculation, its accumulation is higher than the virus. So it has there is a very high amount of the satellite in the plant. Then we checked until 150 days post-inoculation, and then you see that at 150 days post-inoculation, the amount of satellite decreased. So we thought that maybe at the end of the satellite will disappear, because after that it will not be transmitted anymore. So we tried to see if it was transmissible. So we took some plants which were 32 days or 150 days post-inoculation, and this is the virus accumulation alone or with the satellite, at these 2 different days, and we did transmission tests. And we found that the satellite could be transmitted very well, even at the very late stage of infection. So this means that it's not because it decreased in accumulation that it's not transmitted anymore. Then we thought that maybe, we know that in Europe, TYLCV doesn't infect zucchini. But since this beta satellite can overcome the resistance, the gene silencing in the plant, we thought that maybe it could infect zucchini when it is associated with the satellites. So we tried co-inoculation of both, and we did inoculation by mechanical agro-inoculation or vector inoculation. And then we thought that it wasn't possible. So the beta satellite doesn't help the virus to extend its host range in this case. Then we know that tomato leaf curl New Delhi doesn't infect tomato efficiently in our country. So we said, okay, maybe with the satellite it will be better. We tried the inoculation with the beta satellite, and we observed that it was not better. So this beta satellite doesn't enhance the infectivity of New Delhi virus in our country. Then the last thing we did was to see if the satellite could be transmitted from one virus to another virus in a different host. Imagine that a beta satellite arrives from cotton. So it is collected by whiteflies in okra plants, and we know that this virus doesn't infect tomato. So if this virus, Bemisia arrives on a plant which is already infected with TYLCV, can it become TYLCV plus beta satellites? That means that the beta satellite could be transferred. So we tried, and we obtained, in 50% of the cases, the experiment was done twice, and in 50% of the cases we obtained plants which have the beta satellites. Then we say, okay, we have insects which have collected this virus from okra, the virus from tomato, and the beta satellite. If we put it on okra plant, can it transmit the right couples, means Jezera virus and beta satellite? We tried, and yes, we obtained 100% of infection of okra plants. And on tomato, we did the same thing, and we observed that 75% of plants could have the virus and the beta satellite, the TYLCV and the beta satellite. So we were able to dispatch the different couple in the right plant. So this means that the beta satellite can be transferred by whiteflies with the good helper virus. Then, in conclusion, so we have seen that begomovirus of the Mediterranean basin can maintain beta satellites, depends on the host and the satellites. And this has some impacts on virus accumulation and symptoms, depending on the association. And that whitefly transmission is efficient even at the late stage of infection. This means that we have to be very to make a survey, regular epidemiological surveillance in this area, as well in whiteflies and reservoirs, to be sure that we will not have introduction of these beta satellites, because sometimes we may not see them, because they can be there without being seen, and as it happens in Jordan and Israel. And also, what we are afraid is the capacity of beta satellite to adapt to new viruses, because I told you that maybe sometimes beta satellites were not replicated by the virus, but in some case, it could be progressive, and then maybe after a few crops, we will have adaptation and replication by a virus which was not able at the beginning, maybe after mutations and co-evolution of the satellite and the virus. And this has already been shown once, and we have to be in another country, and we have to be very cautious to this. And then beta satellite can be transferred between begomovirus and host plants, even if the virus, which is the helper, the first helper, cannot infect the new host. This means that the satellite it is as if for us the satellite can move alone, because we don't care the virus he comes with. So we are now working on this to see if a virus can even, Bemisia can even inoculate a plant with the satellite alone, and the satellite will wait that a virus arrives. Because we have seen that if the virus is there, it can replicate the satellite. But if the satellite arrives with no infecting virus, can it wait like a component and wait that the virus arrives? So this is what we are working on now. And then what we are also doing is to try to modelize the propagation of a beta satellite in a new environment by using this system, which is a mandatory system with a virus which needs the satellite, and the rest of the environment which doesn't need the satellite, and to see how it can work and if the satellite will be maintained or not. And for this, we will need a better knowledge on beta satellite epidemics in infested area, and we know that in India you have a lot of beta satellites. There is very few epidemic studies because many people are working on molecular biology, functioning of the beta satellite, how they interact with the plant and the virus, but there is almost no epidemiological studies about telling us, for example, this satellite is with this virus and not with this one, and it moves from this plant to this one. We don't have these ideas and this information. This is what we are interested in. Maybe if there are some, it could help to develop, to modelize and have a better knowledge of the disease, of the risk that we may have in the next few years. Okay, that's the end. So my research to the team I'm working with, and this is Montpellier and the seaside. And this are the project that we have worked with, and the Region Occitanie, which paid for a lot of help for students with vectors transmission, and a private company which produce resistant seeds.
Okay, thank you. Thank you very much. We can have one or two questions. Yes, Dr. Karthi.
Did you get any chance to document the alpha satellite?
Yes, we worked on alpha satellites, isolated also from Okra, but we tested 3 alpha satellites, and we didn't have any impact on our virus. So we didn't go further to this. We know that some alpha satellites can produce increased viral symptoms, but the one we have tested did not. So we didn't go further to this.
So far you have not recorded tomato leaf curl New Delhi virus on tomato?
With alpha satellites?
No. Virus, tomato, New Delhi virus.
New Delhi virus?
Did you record in tomato? So far you have not recorded?
No.
Only on cucurbits?
Yes, but the strain we have is different from the one you have.
What is the identity?
The strain we have in Europe, which was introduced in Europe, is called tomato leaf curl New Delhi, Spain. It's a particular strain which was, I think, what I remember, this strain appeared in India and was then introduced to Europe, but the one which is infecting tomato here is not present in Europe. So a particular isolate was transferred there, and this isolate is only mainly efficient in cucurbits. It can infect tomato, but maybe 10% efficiency when you inoculate and no symptoms. But when you add the satellite, you have a little bit symptoms, but not much. It's not a problem in agronomy.
That is by artificial inoculation.
Yes.
Okay, thank you.
Any more? Yes, please.
Thank you, Sika. Just for my understanding, I'm not sure I understood why the viruses that do not need the beta
satellite.
The beta satellites, thank you, create more symptoms.
Why it doesn't need?
When they don't need the beta satellite, why are there more symptoms? Is it adaptive for the beta satellite, or to the contrary?
No, it is because the beta satellite encodes for a gene, which is the beta C1 gene. And these genes, even if you transform a tomato plant with this gene, you will have a viral symptom without virus. So this gene it modifies the chloroplast functioning, and it creates symptoms. So when you inoculate these beta satellites, most of the time you have an increase of the viral symptoms, because this is distortion of the functioning of the chloroplast is added to the viral symptoms, and you have more severe symptoms. But usually the virus infects the plant alone. But when this satellite is present, you have more symptoms, but it's not because of the virus replicates the satellite, so you have the symptoms, but the virus doesn't need the satellite. It produces less severe symptoms, but the virus produces its symptoms, and the satellites add some symptoms to the viral symptoms. So this is something which is associated to the disease, but in fact, if you take a healthy plant and you transform your plant with the gene, you oblige your plant to produce the beta C1 protein, you will have symptoms also. So you understand? So you see it as if it was an increase of symptoms, but in fact it's addition of two types of symptoms that you have.
I think if there is no further questions. One more? Okay. I think we have some online presentation also from Dr. T. Nagan Fon from France.
Yes, I am available.
Yes. Good afternoon.
Yes, good afternoon.
Are you able? Your audio is not visible. Audio is not hearing.
You don't hear me? Good afternoon.
You are not audible.
No, my audio is fine.
Yeah.
You don't hear me?
Please check your audio.
It normally, it's good. It's good.
No.
No problem.
We are not audible. Yeah. Yes, please.
I have such have meeting before.
Laptop amplifier connected. Yeah, it's connected.
Hello.
Yes.
Can you? Yeah.
Yeah. Thank
So,
you, sir.
I think that the problem is from your side, but I have just a meeting 30 minutes ago, and I had no problem with my audio. So, should I share my screen, or the presentation will be on the? I should share my screen now, right? So please give me the authorization for sharing the screen, please. I don't have the permission for sharing the screen.
You have to share the screen.
Yeah, I have to share the screen, please. And the Zoom don't allow me to share the screen. So you should give the permission for me to share my screen.
ஷேர் பண்ணுறேன், நீங்கள் ஷேர் பண்ணணும், அவங்க பண்ணணும். நீங்களும் ஷேர் பண்ணுங்க. அவங்க ஷேர் பண்ணுற மாதிரி பண்ணிட்டு
Currently, I cannot share my screen.
Yeah, please wait for wait.
Yes. Yes. So it's pity that I cannot participate in person because I have some problem with my visa in Cambodia. So I am a researcher from Sirat. So during the waiting time, I can take some time to introduce myself. My expertise is nematology. So I work mainly in the biocontrol of the plant parasitic nematodes in the rice field in the Southeast Asia. And currently, I am based in Phnom Penh, Cambodia, for 1 year to work in the project about the using the microbiome and the bioinoculant against the plant parasitic nematode in the rice field in Cambodia. So I work I work in the team. The team named by BRIO, that stands for bacterial rice interaction and biocontrol. and to share my to share about my research in biocontrol of plant parasitic nematode. I still cannot share the screen.
Can you share from your side?
Yeah. What cannot share now. I cannot share the screen. The heart is not giving the permission for sharing the screen.
இது முடியாதா? இல்லையா? சொல்லிடலாமா நாளைக்கு பண்ண சொல்லி? ஹோஸ்ட் எங்க இருந்து வாங்கணும்? ஓ ஹைதராபாத். I think
time from here?
Wait.
So do you want?
Wait.
Okay.
Wait.
Okay, no problem. Yeah,
There is some technical problem. You please wait for your wait.
But in case you want to skip my presentation to save some time, no problem for me also.
Please check your chat box, and there will be a link.
Okay, I'm checking with the link.
Yeah. So with that link, you join with the Google Meet.
Yeah. So I should click this one to access to camera and video. Yeah.
Yeah, can you share it from your side?
Yes.
Yeah, please.
Sorry. Yeah. Okay, wait. Oh yeah. So do you see my screen now?
Yes, please.
I think you have the duplicate voice in your room because I hear the duplicate voice. Is it okay from the side?
Yeah, please go ahead.
It's okay now? Um, so today
Yeah, you can proceed.
I hear there's a boy I hear my voice, so it's very annoying in my. I will put it out and then when I will stop, I will put the microphone on. So if you have the question, I won't stop because we will have question at the end. Do you agree for that? Because now I hear my voice.
Okay, please go ahead. We are not listening to you. Audio. Please check your audio. Your voice is not coming.
see that you move from the... It's okay now?
Yeah, you are audible now. Please proceed.
Okay. So now you can see my screen, right?
Yes, yes.
Yeah. So, okay, so now I can start. I will do it quick because it takes a lot of time from you due to the technical problem. And today I will present to you nematode that I put the headset because I hear my voice and I cannot. So now about to start the meeting. And I introduced to all of you that I work in plastic nematode that are taking right in my South Asia.
Doctor, headphone oda mic tooki potuchu. Please, your voice is not clear. Can you use your microphone, please? Headphone. Can you please use your headphone?
Plant that can cause annually loss of more than $80 billion. And among plant parasitic nematodes, the melodosine species, also called as root knot nematode, they are only around 100 species, but they are
Please use your headphone. Can you please use your headphone?
They are and they can cause 5% of world agriculture loss, and they account for 50% of total rice production in Asia. And the main character of the plant parasitic nematode, they have the stylet that they can feed on the plant cells. And here are the symptoms of the nematode infection in coffee, rice, and in soybean. And Melodosine gramicola is, as a common name, is rice root knot nematode because they impact mainly on rice production that can reduce the rice production up to 70% in upland and 87% in lowland condition. And the symptoms are very similar with the nutrient deficient symptom with the stun and yellow plant. So they can be underestimated, and with below ground symptom with the gum formation in the root and the rotted root. They can add they live inside the root, and they can induce the gum and can stop plant from can reduce plant from nutrient uptake, and they can attack the right plant at very early stage, and they can cause the plant death and can cause serious impact in the production. And Melodosine gramicula, I will call it MC. They distribute worldwide, and they can increase very fast and across the right growing region. Like in Southeast Asia, account for 90% of rice production worldwide, and the rice production in this area is threatened by MC. And it's very challenging to control these rice nematodes, as they have the rapid life cycle that within few weeks from single juvenile can produce 300 control. And this F of this right nematode is really resilient in the soil that they can stay more than 1 year. And this pest can affect a wide range of parts in more than 120 plant species, mainly in rye, but also they attack the wheat that associate in the rye growing rye agrosystem. So to manage the dead pest in the rye, we also have to manage the dead pest in the wheat. That makes it double difficult. And nowadays, using chemical is become serious problem at the root knot nematode alone. They can accounting for nearly half of nematicide consumption. And the legacy effect of the pesticide when using to control the rye nematode, they can be non-target impact to the other beneficial species, and the residue can impact to the plant health and also human health. And the case study in Cambodia now at rye contribute for 25% of the national economy. And recently, the 1,000 tons of Cambodian rye were rejected from high-quality export markets such as the European Union due to the pesticide residue. So it's an urgent need for sustainable control strategy for this pest. So there are several strategies that can help to reduce the rye nematodes, such as the continuous flooding, water management that can reduce nematode. We can use the organic matter can help to reduce the nematode also, and crop rotation and some available commercial biological control product. And few rye variety are characterized as resistant and tolerant to this rye nematode. However, not any strategy alone they can be efficient to control this rye nematode. There should be an integrated pest management for control of this rye nematode. And in our team, we focus on the biological control of this rye nematode. So to use microorganisms as a biocontrol is eco-friendly and cost-effective. However, for the commercial product nowadays, they are like wide spectra products, and they can target also plant parasitic nematode and also the beneficial nematode. And our purpose is to identify the specific bioinoculum against dry root knot nematode against this species. And other studies they found that there are fungi and bacterial taxa that can have the antagonistic activity against the plant parasitic nematode, such as they can produce toxins, they can induce the systemic resistance of the plant, they can be parasites to kill the nematode, and they can affect or they can kill the nematode at different stage, like from juvenile and female. And we focus on suppressive soil because when before infection to the plant, the nematodes are moving through the soil, and there are soil that can have the suppressive against the plant parasitic nematode. So the suppressiveness of soil can be considered. There are the biotic factor and abiotic factor that can help to suppress the plant parasitic nematode and can promote the plant growth. So for we to study the suppressive soil, our team, the PhD in our team, focus on the different agriculture practice in Cambodia. In Stung Chinis, we focus on the two different agriculture practice, which conservation agriculture and conventional tillage. The conservation agriculture, there's no tillage and there are cover crops. And conventional tillage, we have the frequently the tillage and no cover crop. And and we use the morphological approach and unicon barcoding approach to compare the diversity of the nematofauna and microbiota between different agriculture practice and to assess the suppressiveness against the plant parasitic nematode in different agriculture system, system. And in the study of SPD in our team, we found that the conservation agriculture in Stung Chinit in Cambodia can increase the diversity of the nematode taxa, beneficial of the nematode and microorganism taxa, and importantly, they can reduce the soils can reduce the number of plant parasitic nematodes, and in this case, it's Hitmaniella and Melodozin species. So, the conservation agriculture in Stung Chinit in Cambodia, it can improve the soil quality, and it can increase the rhizosphere biodiversity. It can modify the abundance of trophic weeds, and can allow the maturation of the soil food web. And importantly, they can reduce the plant parasitic nematode abundance. And to test if it's a microorganism that mediate the soil suppressiveness against the right parasitic nematodes. We incubate the soil microbiome with the MC, with the Melodozin gramicula, and we assess the nematode mobility. And we found that there are some the soil from Stung Chinis in conservation practice and in Rovian province with the sustainable practice, they show the strong impact to the nematode mobility that can reduce the nematode mobility that lower than 25%. So this indicate that the microorganism in the soil that derive the antagonistic activity against MC. Sorry. And using the metabarcoding approach, we identify there are more than 100 bacterial and fungal taxa that enrich in the suppressive soil. And among them, there are known antagonists. There are taxa that was known to be to antagonists to have antagonistic activity against nematodes. And among the soil microbiome, when nematodes move through the soil toward the root to infect into the root, the nematode cuticle is highly glycosylated, so the bacteria can stick onto the cuticle, and the cuticle of nematode is the first part that interact with the plant during their infection, and the microorganisms that stick on their cuticle can have the antagonistic impact to the nematode. They can protect the plants from the nematode by they can be paradise with to kill the nematode, or they can induce the systemic resistance of the plant against nematode, or they can secrete the secondary metabolite of volatile organic compound that can act as the nematicide activity against the right nematode. So we investigate. We aim to study if there the microorganisms that are associated to the cuticle of the nematode, and this microbe have impact to the infection of the nematode into the rice, and if the nematode have the plant promoting have their impact to the plant health also. So with the study of our PhD and a master student, we found there are 64 bacterial strains that associated in the cuticle of the Graminicula, and of course we need to include also the endosymbiote, because they also with nematode they have endosymbiote and the cuticular microbe, and we exclude the endosymbiote and we study only the cuticular microbe. And we test the nematicide effect of the cuticular bacteria using the culturable approach. We incubate the bacteria with the nematodes and to assess the mobility of the nematodes. And after incubating nematode with the bacterial solution, we found that there are 9 bacterial strains that can strongly impact to the mobility of the MG juvenile, that they can reduce the ability of moving of Melodogene gramicula to less than 25%. And to sum up for my presentation, in this presentation I would like to point out 4 main conclusions. First, I point out that Melodogene gramicula is the main threat for rice in Southeast Asia, and it's very challenging in controlling this pest. And we found in our study that the conservation agriculture in Stunchini in Cambodia shows soil suppressiveness against this rice nematode, and the microorganisms are the factor that drive the suppressiveness of the soil against the rice nematode. And we found the bacterial strain that associated to the cuticle of the Melodogene gramicula that can have the antagonistic activity against this rice nematode, and this can be the potential bioinoculant for this pest. So the work in this presentation is within the project of my colleague in the team, and my team work as I introduced to you before, that my team work with the bacteria-rice interaction and biocontrol. And the work in this presentation mainly from PhD student from our team and master student. And we are in our team, half of our team are currently based in Cambodia, and our main study are now focused in the rice cultivation, rice agro-system in Cambodia. And thank you very much for your attention and very happy to hear your question and to answer the question. Thank you.
Thank you. I think we can have one or two questions.
Yeah.
Yes.
Thank you again for the nice presentation. I was wondering what are the existing biological control methods against these nematodes, and whether the bacteria that you found, how you think they could be maybe convert one day into a biocontrol method?
Do you mean that the bacteria? Do you mean that is there any the bio product from our? No.
At some point in the introduction, you mentioned a biocontrol, right? And so
Yes.
I was wondering what it is more specifically, and whether your research could transform into new biocontrol methods.
Yes. So the purpose is to find the bioinoculant that can be used as a biocontrol product again, specifically against the plant parasitic nematode in rice. Again, Melodogene gramicula, and we don't want to have the side effect like the other biological. Currently, the commercial bioproduct, they have the wide spectra. They use the wide spectra bioproduct, so this can kill also the beneficial nematode also. So our main target is to select only the bioinoculant again specifically to the plant to the rice nematodes that attack on rice, the Melodogene gramicula. So this is a preliminary resource, and we still have not yet go into detail to test in planta to test in planta for their infection. So in this state, we only test in vitro. We incubate the nematode with the bacteria solution, and we found that certain bacteria can kill a lot of can kill nematodes and can reduce the mobility ability of the nematode. But we haven't tested for the functional. We haven't tested for the in planta for the infection of nematodes, for the impact to the infection of nematode to the plant. And with this, we need more work. We need to go detail in the functional analysis, and to develop into a bioproduct for control of this pest.
Thank you.
Yes, thank you for your presentation. I had a question. Are you aware of the PhD that was conducted up to last year in the Battambang province by Mathilde Dionisi? And if so, she had an experiment on the effect of conservation agriculture on yield buildup of rice and also on pest development. And I was wondering if you know this work, if you would consider designing a new submodel for nematodes, knowing that all the crop model part and other diseases and pests are already designed, and it could be an outcome of the work that you are conducting. What is your feeling about that?
Yes. So I know Martin.
Sorry, we cannot hear you.
Really? I know Martin, and we work with the team in Battambang also.
Oh, sorry, Can you fix the sound? Because before it was loud enough.
In my side, there was no problem. There is no problem in my side.
Yeah, but here we can read your lips, but I'm not sure to really understand.
Yeah, can you please adjust your mic?
Hello.
Little closer.
Hello.
Yeah, you are not audible much.
You don't hear me?
Yeah, we are not hearing you.
Okay. It's strange.
I think you can send in the chat box. If you don't mind, you can
Yes.
get it. Because we have given you sufficient time. We are running out of time. Thank you. Thank you so much for your presentation. I think with this we can come to the closer of this session. Since morning, thank you very much for all the speakers. Excellent presentation by all the invited speakers. I think I request all the speakers kindly share your 2-3 points which we can bring into the proceedings. Of course, we will be finalizing the proceeding, but still we require from the speakers. That will be more valid than what we propose. Perhaps the projection for the new programs from the French, and similarly, you people also, you propose from your side what you expect from India from your presentation. So how best we can collaborate your partners. So that should be the highlight of the proceedings, that perhaps that will help us to bring out the final the preliminary session discussion and all. I think with this request, I once again thank all the speakers. Before I close, I request our co-chair to comment.
Oh, I don't have much to say. It was a pleasure to to chair this German session. So you hear very nice presentations, and it was quite interesting. So I hope that we will develop nice projects from this. Okay.
So before closing this session, I have, because I didn't ask anything from any speakers, so I observed. So in the nutshell, I want to put a few suggestions. It is up to the speakers to think over. One is that from the presentation of Dr. Kumar, kindly let us try to explore the cold tolerant or the cold temperature tolerant microbiome. That is the one unexplored area, whether it is in the northern hill or southern hills where the temperature is very low, but the crop, there are very high value crops. So that we can think of. And similarly from the modeling, I think we have to strengthen the modeling, particularly for the crops like potato or the vegetable diseases. So we can come out with a very good modeling program. And for potato exclusively, the PCN, potato cyst nematode, and the lead blight combination. Because these two biotic stress are very crucial for the hilly potato cultivation. So this we can think of. You can integrate with the pathology, and we can take the support from the nematologist from the French, and we can bring out some program. And another very important observation, what I heard, that the most of the hilly regions we have the high value vegetables, particularly the beans, where the Xanthomonas disease was discussed. And I think most of the beans cultivated in hilly areas, I think a lot of beans collection, a lot of germplasm we can explore. Perhaps we can give our good germplasm materials to the other country people. They can evaluate our material, and we can also take their product, and we can develop a lot of the genetic resources. Already the genetic resources are available in the hilly region, particularly in the Nilgiris, lot of beans. The local people are maintaining a lot of seeds. I think the vegetable scientists may be knowing this, and we can explore it maybe through our NPPGR, we can explore. And biofortification. The vegetables are lacking, vegetable groups are lacking for the biofortified vegetables. Of course, we are concentrating on the cereals till date, and we can go ahead with the vegetables also. Maybe some good vegetables are there. There are some vegetables which are having the medicinal values. So those things can be explored. So this is my personal observation. So with this, I thank all the participants, all the speakers, and the organizers for giving this opportunity. Thank you all.
So this is for the participants from outstation and friends. So if you have any idea of exploring the city for shopping and all, we can arrange the vehicle. Otherwise, we can wind up the today's session. Okay. So tomorrow's session started.